Hey, this is a video for Respiration 3, so go ahead and check out the essential vocabulary and set up your guided notes. And let's look at the aim. Explain how carbon passes through plant and animal cells during cellular respiration. So the mitochondria are these organelles. They're the organelles in both plant and animal cells that perform cellular respiration. The mitochondria, you may have heard it called the powerhouse of the cell. While it is true, that is not a description that we use to describe it, because you can be more accurate by saying it produces ATP, it performs cellular respiration. Now, plants and animals do respiration slightly different. So I want you to tell me which organelles are involved with plant cell respiration. So I'm going to walk you through this. First of all, we have carbon dioxide entering through the cell wall and then the cell membrane. It's going in through the cytoplasm, cell wall, cell membrane, into the cytoplasm, and then into the chloroplast, where it is converted into glucose. That glucose then travels to the cytoplasm and then to the mitochondria, where it is converted into ATP. Now that ATP is used throughout the cell. The ATP that is produced, the cells are using that ATP within that cell. So in your notes, I would like you to tell me which organelles were involved with plant cell respiration. Now with animal cell respiration, I want you to tell me which organelles are involved. Now remember, animal cells are not producing their own glucose. So the glucose has to enter the animal cell through the cell membrane. Then it has to enter through the cytoplasm, where it goes into the mitochondria. And in the mitochondria, ATP is formed. So in your notes, I would like you to indicate which organelles were involved in the animal cell respiration. Please indicate that. Now, one experiment we could do is to determine the impact of glucose, or the inputs, on the rate of respiration, or the output. So we can't necessarily measure how fast respiration is happening, but what we can do is we can measure some of the waste products. So we can adjust the glucose and we can measure the amount of carbon dioxide that's produced. One way you do this is by using an Erlenmeyer flask and using some yeast. Yeast are small organisms, small fungi that basically yeast is what you use in bread, but they will metabolize the glucose or the sugar and they will produce carbon dioxide. So we can have different concentrations. We can have one with 0% glucose, one with 2%, one with 10%. Put a balloon on top of each one. Let it sit for a while, and you'll notice that the 2% glucose got slightly bigger, and the 10% glucose got even bigger. This is because this first one produced no carbon dioxide because there was no glucose. This one produced a little bit of carbon dioxide, that's why the balloon filled up slightly, and this one produced more carbon dioxide because the glucose rate was higher. So if you increase the glucose, respiration rate will also increase. So what is the hypothesis that is being tested in this experiment? Please indicate that in your notes. Now for the check for understanding, I would like you to respond to the aim. Explain how carbon passes through plant and animal cells during cellular respiration. And complete practice problem set 3. I hope that this video is informative. I thank you for your attention. I'll see you soon.